I appreciate the opportunity to present today. The title of my presentation today is Advancing Cancer Therapy Harnessing in Vivo Tumor Models for Breakthrough Drug Development. I'll be covering the value and applications of different tumor models and how our company uses these models to advance preclinical oncology research. The presentation will be break down to the following three parts. Firstly, the background and applications of tumor models. Secondly, the capabilities and case studies of tumor models in GenoBioTX. Thirdly, I will introduce our company and our capabilities in oncology study. I'll begin by the first part. This is a brief background of the preclinical mouse models and clinical trial in the drug development. Preclinical mouse models are essential for developing new cancer therapies. They help us identify and validate new cancer drivers and therapeutic targets, analyze drug pharmacokinetics and pharmacodynamics, assess the anti-tumor efficacy of new drugs in vivo. This model bridges the gap between basic research and clinical trials. Once we obtain promising preclinical data, candidate drugs will move into human trials to evaluate safety and efficacy. However, some patients may experience poor treatment due to intrinsic or acquired resistance. By using preclinical models, we can explore these resistance mechanisms, identify predictive biomarkers, and develop combination therapies to enhance treatment outcomes. The close integration of mouse models with human clinical trials enables better patient stratification, discovery of new biomarkers, development of effective treatment and strategies, ultimately leading to improved patient care. For over 50 years, tumor transplantation models using nude mice have been essentially used in cancer research. This model typically involve either transplant human cancer tissue derived cells, xenografting, or mouse tumor cells, allografting. Their major benefits is that they are cost-effective, offer synchronized tumor growth, and are relatively simple to work with, and also there are a lot of cell line resources are available. However, these models also have some drawbacks because of their rapid growth rate and the non-orthotopic nature. They often do not replicate the natural tumor environment accurately. The tissue structure and the microenvironment can vary significantly from those seen in the actual tumors. Actually, long-term cultivation in vitro can reduce the genetically diversity and cause permanent changes in gene expression, making them less reflective of true tumor biology. Then we introduce the patient-derived xenograft model. This is typically generated by subcutaneous implantation of fresh surgically derived human tumor material into immunodeficient mice. This approach preserves the tumor's molecular and genetic information, maintains tumor tissue structure and microenvironment, and is highly effective at predicting therapy responses, making it a powerful tool for personalized medicine. However, PDX models have their limitations. They are challenging and costly to create, require spatial immunodeficient mice, and show variable engraftment rates depending on the mouse strain and the quality of tumor sample. Engraftment success can also differ widely across tumor types and over time. The human tumor stroma is often replaced by mouse stroma, adding to the high maintenance costs. To be noted, our company has established an inventory of more than 5,000 tumor tissues across different cancer types. Another type of tumor models are genetically engineered mouse models, also called GEMs, induced by oncogene overexpression or tumor suppressor gene depletion. They can introduce specific mutations, grow tumors in a natural setting, and predict drug responses well. However, they also have drawbacks. They may skip key cancer stages, making them less representative for some research areas, such as metastasis. They are also expensive, time-consuming to create, challenging to scale up with uniform growth within a limited time. In short, the GEMs are highly accurate models, but their cost and complexity can be limiting. We will also introduce a series of GEM models in this webinar that showed in our company. Later on, I would like to introduce the current application status of different tumor models. Here is a systematic survey of about 950 studies published in 2016, conducted to quantitatively review the status of tumor model experimentation. From this table, we can see that 80% of the in vivo studies rely on cell line-based models. More advanced models like gene models, environmentally induced models, and PDX models are not that used frequently as CDX, and their availability is depend on the tumor type. Let's take a closer look at the breakdown of application of cancer models. Panel A shows the model usage. The pie chart shows that 82% of studies just use a single model, and 89% of studies focus on a single tumor type. This tells us most research is pretty focused, sticking into a narrow scope. From panel B, we can see the model distribution by tumor type. The bar chart shows that breast cancer is the most studied, followed by lung and colorectal cancers. Meanwhile, cancers like liver, thyroid, and cervix are studied far less often. From panel C, they summarized the model preferences by cancer type. The bottom section shows that the red circle indicates the cell line derived. Green is the PDX. GEM shows red, and environmental induced shows blue. The CDX models are the top choice of the most cancers, making up 93% in breast cancer and 85% in lung cancer. PDX models, as shown green, are more frequently used in pancreatic cancer and melanoma. Genetically modified animal models, as shown yellow, stand out in the colorectal and pancreatic cancer, while the environmental induced models are rarely used overall. In summary, cell line models are still dominate, but PDX and GEM models are becoming more prominent, especially in cancers where understanding genetically and environmental factors is crucial. That's the background and the current status of tumor models. Next, I will introduce some case studies of tumor models that applied in drug efficacy study. Let's briefly go over the different types of tumor models. The syngeneic models. These include syngeneic models, xenograft models, and spontaneous tumor models. Syngeneic tumor models involve tumor cells derived from the same species as the host. For example, mouse tumor cell lines injected into mouse. They are ideal for studying immune response and cancer immunotherapy in a fully functional immune system. Our company can run both subcutaneous and orthotopic transplantation and metastatic tumors. Xenograft models include CDX and PDX models, where human cells or tissues are injected into immunodeficient mice or rats. This category also includes the human immune system reconstruction models, which allow for testing in a human immune environment. Lastly, the spontaneous tumor models. In these models, in this model, tumor arise naturally due to genetical modifications, such as using some oncogenes. Examples including KRAS LKB Luke mice, KRAS TRP 53 Luke mice, KRAS EGFR spontaneous lung cancer, and KPC spontaneous pancreatic cancer. After the tumor was developed, we can isolate the tumors, develop them into immortalized cell lines for further xenograft or syngeneic study. With our gene editing expertise, we can modify cell lines, mouse and rat models to humanize specific targets based on project needs. Further, later on, I will introduce one case study for these categories gradually. Here is an example of a syngeneic model. We are looking at the in vivo efficacy evaluation of immune checkpoint inhibitor in the treatment of MC38 tumor model in C57 mice. We have two groups. One is vehicle, another one is treated with anti-mouse PD-1. The key findings is that you can see a clear reduction of tumor growth in the anti-PD-1 treated group, showing the treatment effectively inhibits the tumor progression. The right graph tracks the body weight, showing no significant body loss, which means the treatment is well tolerated. So this study highlights that the checkpoint inhibitor not only suppresses tumor growth, also has no major adverse effects, and also suggests our model is well validated. Then next, move to human immune system reconstruction model. NMG mice is a mouse model, is a highly immunodeficient models that lack T, B, K, NK cells due to mutations in both PRKDC and interleukin-2 receptor G genes on nude mice. This is independently developed by our company. The bar charts on the right compare immune cell populations across different mouse models like BOB-C, NOD-SCID, and NMG, and shows data in both spleen and blood. You can see the NMG mice, as shown green, has has a very low percentage of T cell, B cell, and NK cells compared to other strains, making it a robust platform for human immune system reconstruction. This slide introduces two types of humanized immune system reconstruction. The first one is human PBMC reconstruction mouse model. This is created by injecting human PBMC into adult NMG mice. After 3 to 4 weeks, the mice develop a human-like CD immune system, making them suitable for studying infection disease, gene therapy, hematopoiesis, drug targets without cross-activity, and immunogenicity study. Another type of model is CD34 HSC reconstruction. This is developed by injecting human hematopoietic stem cells into newborn irradiated NMG mice. This model takes 12 to 16 weeks to fully establish and is ideal for long-term studies involving cancer research, immuno-oncology, and other therapies requiring a stable human immune system. This slide compare these two models. From the left, we can see the PBMC humanized model is quick to establish. It can create efficient T-cell engraftment in just 3 to 4 months, make it ideal for short-term studies. However, due to high presence of T cells, it's prone to have a lethal GVHD effect, limiting its use in long-term studies. For the humanized SHC model, takes around 12 to 16 weeks to develop, but this model offers more balanced immune cell populations, including T cell, B cell, and myeloid cells. There is no GVHD risk, making it suitable for long-term studies. It is a preferred model for researching research involving cancer immunotherapy and other applications needing a stable human immune system. Both of the human PBMC and CD34 reconstruction have its advantages and disadvantages. In this slide, I will introduce how we select a donor for the humanized PBMC reconstruction model. A good donor will show rapid immune cell reconstruction and a low risk of GVHD. In fact, in the left paragraph, we injected PBMC to 6 different donors. Donor 1 and donor 6, as shown, the red and orange, show the very poor CD45 cell reconstruction rates, so they were excluded. From the right, we can see donor 4 has caused a noticeable weight drop, indicating poor tolerance. So we exclude this well. 2, 3, 5 finally will be selected for future experiments. This is another example. From the left paragraph, you will see donors 1, 2, 3 show the good immune cell CD45 cell reconstruction rates. From the right paragraph, we can see donor 3, as showed as green, caused a significant body weight drop, indicating poor tolerance. So we excluded. We choose donor 1 and 2 as optimal candidates for future experiments. Usually, we purchase and store a complete batch from the selected donors to maintain a lot, keep a consistency across multiple experiments. The next 2 slides will show 2 case studies of humanized PBMC reconstruction model in efficacy study. looking at the schedule, PBMC was engrafted 2 days before the tumor engraftment. Then treatment began on day 8 and continued at regular intervals up to day 21. From the findings, we can see top left graph shows successful PBMC engraftment and a steady body weight. The top left, the bottom left shows the tumor growth is very stable over the time course. So this is a good donor for this human reconstruction model. Then we use a test antibody. We test an antibody in this model and found the test antibody could significantly reduce the tumor volume in 17 days. This study shows this is a good model. This is a good model this is a good human reconstruction model for the drug efficacy. Here is another model called PBMC reconstruction based on MHC B2M double knockout in MG mice. We created this mice is to reduce the GVHD effect while still maintain the effective human immune system reconstruction. Of the tumor growth data on the left shows treated groups show significant tumor inhibition compared to the control, while body weight remained stable, indicating good treatment tolerance. The right panel shows the status of immune cell population during treatment over the time course. Overall, this model is also ideal for long-term studies that need robust human immune system with minimal GVHD risk. Later on, we will introduce some case studies for HSC reconstruction. The top panel, after injected with 30 CD34 cells, we detected the human CD45 in the blood marrow and over the time course. It shows successfully human immune system reconstitution. And we from the right and the bottom panel, we can see the populations of T cell, B cell, NK cell, human monocytes, and dendritic cells also showed substantial amount. To support, additionally, to support the NK cell development, we create in human interleukin-15 and MG mice, then inject the human interleukin-15 to support the NK development. Compared to the regular CD34 model, the NK treated group showed a much higher percentage of CD55 cell population. And the right flow plots demonstrate enhanced presence of mature NK cell population markers, such as CD16 and NKG2D, confirming successful NK cell development. This model is ideal for studying NK cell biology and testing therapies targeting human NK cells, providing a robust platform for immuno-oncology study. Then move to case studies. This case study showing shows the HSC reconstruction with a test antibody. We can see the test antibody result a significant tumor volume reduction The right paragraph showed the initial stability of body weight, but later declines, suggesting potential tolerance tissue issues with antibody. So we show another case study, test antibody 1 and 2. While test antibody 1 doesn't have any inhibition effect on the tumor volume, while test antibody 2 could greatly inhibit the tumor growth without any change of body weight. This study, these case studies shows the HSC model as is a reliable platform for evaluating antibody efficacy and safety. Based on previous studies, we know that the timing of PBMC injection or treatment is very critical. Too early or too late can impact the treatment window and overall efficacy. Our company has extensive experience in optimizing these conditions for robust study outcomes. Here is a list of well-validated cell lines based on the NMG mice on various cancer types, lung cancer, leukemia, pancreatic cancer, etc. If you have any other questions in the future, please do not hesitate to email us. We are happy to schedule a meeting to discuss. Another part is the orthotopic and metastatic tumor models. Even if subcutaneous models are easy to work with, because the tumors are visible and can be measured with capillars. However, the orthotopic models are more realistic. They can better replicate the natural tumor environment and disease progression, making them highly valuable for preclinical studies. The downside is that the orthotopic models are more complex to establish, often requiring bioluminescent-based labeled cell lines for imaging and precise techniques for injection, which can lead to a lot of variability. Our facility is well equipped to handle these challenges. We have extensive experience with orthotopic injections and imaging-based tumor measurements AVIS system. Plus, we have a large inventory of luciferase-labeled tumor cell lines to support this research technique. Here is a summary of validated orthotopic and metastasis models developed by our team, covering cancers like lung cancer, pancreatic cancer, hepatic cancer, mammary pad, glioma, renal cancer, cervical, and metastasis cancer, hepatic cancer, and pulmonary metastasis, and formal injection for bone metastasis. This is a case study shows the lung orthotopic tumor model where tumor growth was tracked by a luciferase labeled line. The right panel shows the imaging via IVs over time. The left panel shows the data, the total luciferase flux is gradually increased over time. Here is another case study showing the liver metastasis model via spleen injection. We have successfully completed a couple of orthotopic projects for our clients. That's all about the transplantation models. It is noteworthy that we have various spontaneous tumor models, including mutations such as KRAS G12D, G12C, TRP53, KPC, among others. For majority of these models, we have detailed validation data. We can show you if you are interested. Just email to us. On this slide, we have a case study using showing the KPC spontaneous pancreatic cancer model and derived cell lines. This model is created by crossing KRAS, G12D, TRP53, and PDX3 mice. This model results spontaneous pancreatic tumor model that we need to closely monitor them because the mouse are very fragile. They tend to die in 100 days. The right panel, after the tumor was developed, we take out the tumor develop into an established cell line, then perform an efficacy study after subcutaneous injection into subcutaneous this cell line into C57 cells. After inject into a C57 mouse line, this treatment caused an 78 reduction of TGI and in the end point. This is another cell line, KRAS LKB Luke, developed from KRAS LKB Luke mice. After the tumor was developed with AAV treatment for 2 to 3 months, tumors was isolated, then cultured into cell line as shown in the left. Here is the picture of the lung and the cell line. Then we tested the cell luciferase intensity. 3 of the donors shows very strong luciferase. Both of these 3 shows very strong luciferase activity, and the tumor growth data also shows that 3 dosage of cell inoculation all cause a stable tumor growth in this in the mouse model. This also suggests the KRAS LKB Luke is a well-validated cell line to perform efficacy study. Apart from these developed cell lines, our company also developed a lot of humanized cell lines based on different tumor types to support immuno-oncology study. Here is a list showing partial of the cell lines. For example, MC38 cell line, CHO, A20, B16, F10, etc., with different humanized targets humanized. This model especially useful for testing a wide range of therapies, including nucleic acid drugs, ADCs, etc. That's all about the tumor models I would like to introduce today. In the last part, I would like to briefly introduce our company, GenoBioTX. We are a well-known CRO company which aim to provide both pharmaceutical companies and academia clients with professional, comprehensive, and convenient preclinical research services. Our standard model inventory includes genetically engineered mouse models, which use for genetic function research and drug target discovery. We also have disease efficacy evaluation model, which include immunodeficient mice, humanized mice, immune cell depletion mouse model, spontaneous tumor model, and a genetic disease mouse models for target. All of this can be used for target validation, drug safety testing, pharmacokinetic studies, and more. Here is an overview of our core products and services. Look at the bottom. We have established more than 20,000 strains of models for clients, and our commercialized mouse strains is more than 10,000. Our annual model creation capacity is more than 4,000, and our animal facility can house more than 160,000 cages of mouse. And our company also can provide a lot of services followed by the model development, including model customization, breeding, and pharmacology services. To wrap up, I would like to summarize by our tumor pharmacological platform in GenoBioTX. We provide a syngeneic mouse model. We can run syngeneic models, CDX, PDX models, PBMC, CD34, HSC models, spontaneous tumor models that backed up by cutting-edge micro-CT imaging and the IVS system. And we can also support IND filing in both United States and Europe. Additionally, we can provide cell viability assays and a lot of in vivo and in vitro mechanism analysis backed by the by multi-skilled facilities, as shown on the right. That's all about today's presentation. Thank you for your attention. If you have any questions, please do not hesitate to put it in the chat box down below or ask me after this webinar via the email info@genobiotx.com.